Great, Dennis. It's amazing how one person like you could oversee such a broader a range of activity. You must have about a 150-hour work week. That's.
Oh, I don't know what's going on.
Well, no, I think you know what's going on very, very well. That was the best overview of just everything going on in polymer nanopolymer manufacturing that I've yet heard. So, kudos to you. Really well deserved. We are running a little bit late, so I'm just going to try to scrunch down our Q&A period here to something between 5 and 10 minutes. And but we've had some fascinating presentations, so I invite any of you to approach the microphone, introduce yourself, and ask a question of either Rutledge, Russell, Alexei, or Dennis. They're all starving. Let me start off with one question. I'm curious if maybe this is a question, Dennis, to yourself and to Russell. Do you see any kind of overlap between the sort of nanoparticles that Russell's company and academic efforts are producing for pharmaceutical use in his nanoengineering template, and the more broad and not necessarily organic efforts that you have going on at your university and your company?
Yeah, I think what the synthetic organic polymer chemist brings to the table is just a lot of decades and a century or more of organic chemistry. And what's fascinating is these are the kind of conferences that I love to go to because there's none of my colleagues here. I think I know 2 people at this conference, and we typically go to polymer workshops and polymer symposia, and we're all talking about similar things. We're all competing, and the actual customer, for lack of a better word, to go on downstream is not there. So I love to go to the optical conferences and go to these. So I think there's just tremendous opportunity with, I just showed you one of Joe's slides that had 9 different commercially available monomer-derived polymers, which he's made these nanoparticles out of. So I think when people like these gentlemen sit down with somebody like me, then suddenly the blinders are off, and there's lots of very rich opportunities.
Great.
Yeah, I guess I'll just add to that, and maybe I alluded to this, it's very frustrating for people, I guess like me, that are more translational, that are looking to get things into humans, but have the realities of regulatory bodies. Because it's clear that if you're looking at a new chemical entity, silencer RNA, a new drug, and you're thinking about 15 years and $1 billion on average, you would like to not head down that pathway with a new structure, a new material, because that's going to add to the complexity and the cost. But the reality also is, from a science perspective, is I think that's the future of nanotechnology. And so we're trying to practice nanotechnology using existing materials that the regulatory agencies have already seen, but that's not the future. I think the future and the exciting aspects are a lot of the technologies and materials, and I hope that we can bridge that. Again, but the reality is that ultimately we're not the deciders of that. These have a process of approval that they have to go through, and just thinking about people have mentioned polylactide glycolide, which is one of the first bioresorbable materials. The length that took to get approved. Now, we've learned a lot from that, and many other, I think the process is much easier now because of that, but there is a disconnect.
And another question I think maybe in some people's minds is, as they listen to Rutledge's presentation and they see that he's doing some very trailblazing work, again with novel self-assembling gels and whatnot that are being used in surgery, nanotechniques for neuroscience and neurotechnology. He's going between Hong Kong and MIT. Now, of course, the FDA has no jurisdiction over what goes on in Hong Kong, and I'm wondering if there is some way to sort of perhaps lead by example that some of the great work, Russell, that your company is doing, perhaps the part that is using grass materials, generally recognized as safe materials, can be conducted in the U.S., and what you see as the next generation materials, the ones that the FDA has not yet really provided you any clear guidance on or any safe harbor within which to go into man, that perhaps those activities could occur in jurisdictions such as where Rutledge is operating. And I don't know, Rutledge, if that was part of your thinking in working there.
There's a couple of reasons why I went there, but let me just back up just a second. There's a couple of distinctions that I think need to be made, and they haven't been made yet. One is what I'm using is it's classified as a medical device because the breakdown of the material is not creating a cascade that is causing any therapeutic effect. Some of the materials that these guys are talking about actually have a therapeutic effect, so they may be combination devices. So let me just say that to start. But one of the reasons I went to Hong Kong, aside from funding issues, was in Hong Kong and China, there are a lot of people, and in a lot of cases, to get approval to do some of these things, if you can show that they're safe, you can go ahead and do a clinical trial with just an IRB approval or a very small tox and safety trial and loading trial. And one of the things that we have been doing is we've been looking at these materials because this first sequence is actually in humans already. And because it's part of the genome, it's actually being produced by the human genome. It's also produced in other organisms as well. And the breakdown of every one of those different amino acids actually doesn't cause anything. So, and I alluded a little bit to that when I was talking about the spinal cord work. The pH is an issue. And in a lot of the controlled release drug delivery materials, there's a pH breakdown, and it actually goes acidic. And there's been several instances where some of the cancer controlled release materials, not nanomaterials, but regular materials, have actually reduced the pH, which has caused a lot of pain. And there's some work that's recently been done where the breakdown is either neutral or slightly basic, and that pain is no longer associated with that. I don't know if you guys have seen any of that work at all. It's not my work, it's someone else's work. But when I started this, I was concerned about pH breakdown. And in the spinal cord, since the neurosurgeon did it, he cleans everything up, there was no buffering effect, and we saw that. So we saw death of some of the cells. So I think pH, and we saw it again with the AFM data that was just presented that was beautifully done. pH is a much bigger issue than most people think, but we think about pH as a large measurement as opposed to a nanodomain measurement. But I'm not answering your question. Let me answer the question. Yeah, I'm in Hong Kong not only because of the facilities and things like that, but also for access. I'll give you one example. The Spinal Cord Clinical Trial Network is throughout China. I've been through 22 hospitals all over China. In one hospital, in the US, for a spinal cord trial, you want to enroll 1,000 patients. Figure it's going to take 15 years, minimum, because you want very narrow criteria. In 6 months, you can enroll 1,000 spinal cord injured patients with very narrow criteria in one hospital. In 6 months. Now, we all know patents last 15, 17, 20 years. So if you think about the duration, the time value of money, even if it's a little bit more expensive, where you can afford to actually pay for all of the care, you can actually get the trial done much quicker there. And I also want to make this one comment. I do not look at other countries as guinea pigs for the US. I look at it as a way to cure people in the environment they're in and using resources from outside of that environment that may bring care to people who wouldn't be able to afford it. It's very different, and you'll hear a lot of this in the future. And the other thing I will say about China is everyone, we're blessed because we all have healthcare insurance. In China, it's almost 100% pay-as-you-go medicine. So if you have a stroke and it's going to cost 100,000 RMB, the people get together with the other villagers and they decide whether or not they can afford that, or they can afford 40,000 RMB. That's how much care the person gets. For 2 years of care, treatment, surgery, decompression surgery, and stabilization, as well as therapy for 2 years, it costs about $5,000 US for one patient, just to give you a few economics. But I know it was long-winded, sorry.
No, it's a great explanation. I think you brought home the point that a lot of things can be done outside the U.S. for the benefit of the people outside the U.S.
Right.
And perhaps that will become if I could borrow Alexi's term, a forcing function for the FDA to provide more of a safe harbor for Russell's research and for the research that Dennis's technology is enabling going forward. Well, I do hear a few grumbles of the stomach. So the next item on our agenda is lunch. It's 12:53 now, and originally we were supposed to we had allocated ourselves 45 minutes for lunch, but we are running a little bit late, so I'd ask if everybody could have a bit of a quick buffet, and if we could all meet back here at 1:30 to have Dr. Blumberg's keynote address occur at 1:30, and that's what you call a working lunch. See you back here 1:30 sharp.